Thank you very much, Professor Chaudhary. It's really an honor for me to be called upon to deliver this talk at IIT Jodhpur on the National Science Day. I've been listening to the talks by Professor Chidambaram and Professor Brahmachari. I would not have such a broad sweep of science and technology post-independence and pre-independence as well. But I'm going to be extremely focused on a particular domain of science where I have made some contributions, and I will chart the history of that particular domain and inform all of us what may have been the impacts of that kind of work on our understanding of India and the population of India. So I've titled my talk as Celebrating Human Diversity in India, and I'm going to chart the legacy of Prasanta Chandra Mahalanobis through this talk. The first point that I want to make is that we are talking about diversity, and diversity essentially means differences. That diversity would not exist if there were no differences. And I'm going to talk about human diversity. So I'm talking about differences among human individuals. The fact is that when we talk about diversity, human diversity, it really belongs to us. Sometimes we view diversity as though it's alien to us, but that's not true at all. It belongs to us, and we must learn to appreciate and share diversity primarily because it helps unite and educate us. If we understand what diversity is, what we can learn from others, and we can actually improve ourselves, and that's one reason why we should celebrate diversity. The second reason is that understanding people and their backgrounds is crucial to personal and community growth. And there is cooperation builds, respect builds towards other people, other communities, etc. And therefore, the lack of understanding does not promote this growth. And therefore, it is important for us to understand and to celebrate. Today is a chance to find out more about what we have in common rather than what separates us. Oftentimes, we try and think what separates us, and that's a reason for a lot of strife. But if we try to find commonalities in the midst of all of these diversity that we have, that really helps in both personal and community growth, and also promotes peace and harmony among communities and among individuals. So that's the reason why we should celebrate. This is my explanation as to why we should celebrate diversity. I'm sure that other people will have other kinds of explanations as well. The whole idea of studying diversity actually stemmed, or studying diversity systematically, stemmed from this person who actually established the Indian Statistical Institute, and also is the father of statistical sciences in India. He was a firm believer that scientific and rational views lay the foundations of modern age. Prasanta Chandra Mahalanobis was, of course, preceded by a Britisher whose name was Herbert Risley. Risley actually made a lot of measurements of people of India, but that was primarily from the point of view of quantification, and it was very descriptive in nature. Risley actually did not set out to answer specific questions, which Prasanta Chandra Mahalanobis did, of course, not alone, in collaboration with a very famous anthropologist whose name was D.N. Majumder, and I'll come to that in a minute. The Indian Statistical Institute also has as its motto unity in diversity. And for those of us like me who grew up in the Indian Statistical Institute, we are a firm believer that in diversity there is unity. And therefore, we try to look at common threads. We try to find unifying heritage in the midst of all of this diversity. And that's essentially what I'm going to impress upon you. It's also interesting, in view of what Professor Chidambaram has said and what Professor Brahmachari has said. This is something that's not known. That the Indian Statistical Institute, when the Indian Statistical Institute was set up in 1931, there was no Institute of Statistics in the world. Well, there was in England. There was no Statistical Institute in the United States of America. The Indian Statistical Institute was adopted as a model for the first Institute of Statistics that was established in the US at the Research Triangle Park by Mary Cox. And this is a matter of great pride, not just for Indian Statistical Institute and PC Mahalanobis, but also for all of India. And this is not very well known. So I thought that given what Professor Brahmachari and Professor Chidambaram have said, I think this is something that's significant. And we need to also be proud of the fact that the Indian Statistical Institute was adopted as a model in a developed country like the United States. Just to tell you a little bit about Prasanta Chandra Mahalanobis and how he got into all of these diversity kinds of activities and how he set up the Indian Statistical Institute, because I've been told that today we also need to look at legacy and look at legacy of our scientists. So I'm just concentrating on one scientist, PC Mahalanobis. In 1912, he passed BSc with honors in physics from Presidency College in Calcutta and left for England to study at the King's College, Cambridge. He got his tripos there and returned home for a short vacation and never went back. Of course, the reason that he never went back was because the war intervened and he was unable to go back. At that time, he was asked to teach physics at Presidency College, and he was indeed a physicist by training. He became a professor of physics at Presidency College in 1922 and served until 1948. Just before he was leaving Cambridge and boarding the ship, his tutor in King's College, Macaulay, drew his attention to this journal called Biometrica. Biometrica was then the journal, is the first journal in statistics. And of course, statistics grew out of these various kinds of biometrical measurements that people had done. And one of the foremost people who had systematically did biometrical measurements on humans and other animals and plants was Carl Pearson. And Carl Pearson's name I highlight primarily because his name will come up again during my talk. Mahalanobis found some of the papers when Macaulay gave him one volume of Biometrica. He found some of the papers in Biometrica in the journal rather interesting, and he bought a whole set of volumes on his long ship trip sail to back to India, and he bought this entire set of volumes of Biometrica that was available and studied all of the papers during his long voyage. He formed the statistical laboratory in Presidency College that was to eventually be established in 1931 as the Indian Statistical Institute. And that's a picture of the Indian Statistical Institute. When he joined the Calcutta University, there was a department of philosophy. There still is. There's a department of philosophy, and Acharya Brajendranath Seal was the head of the department of philosophy. Acharya Seal was a polymath. He had a very broad vision. He knew a lot of subjects. And one of the things that he told Prasanta Chandra Mahalanobis is that Prasanta, you have to do work in India similar to what Carl Pearson has done in England. I found this note in the archives of PC Mahalanobis, and this is really a translation. The note was actually written in Bengali by Brajendranath Seal to Prasanta Chandra Mahalanobis. So Brajendranath Seal was already aware of Carl Pearson and his work in England. And again, to remind you, Carl Pearson was the first editor of the journal Biometrica. In 1920, Mahalanobis went to the Indian Science Congress that was held in Nagpur. And there he met Nelson Annandale. That was to change his life forever. Mahalanobis' life forever. Nelson Annandale was the director of the Zoological and Anthropological Survey of India. One of the things that during the discussion that they had in the Indian Science Congress, they tried to figure out the Anglo-Indians, which were really meetings between the native Indians and the Britishers. What were they closer to the Britishers? Were they more similar to the Indians, and so on and so forth? So they thought of some interesting kinds of questions on the Anglo-Indians, and Mahalanobis set out to answer those questions by measuring physical dimensions of the Anglo-Indians of Calcutta. And it was a series of papers that were published in the records of the Indian Museum. If not for anything else, this laid the seeds of the growth of statistical science in India. It did not, I don't believe that these studies on Anglo-Indians really impacted very much on later human genome diversity studies, but it did lay the foundations of statistical science, and it also brought to the fore some questions that were later asked during the genome diversity studies and other diversity studies, human diversity studies in India. In 1925, Mahalanobis, a statistician, was invited to deliver the president's presidential address in the anthropology section of the Indian Science Congress. And he titled his lecture as Analysis of Race Mixture in Bengal. And he, of course, based this very much on the studies that he had done among the Anglo-Indians and the kind of questions that he asked. And this is just a sampling of two questions. Do the Anglo-Indians show greater affinity with the higher castes of Bengal or with the lower castes? In other words, did the meetings take place between the higher caste populations with the Britishers or the lower caste populations? That would show the affinity, measurement of affinity would provide an answer to this question. Is there any appreciable similarity with the aboriginal tribes of the Anglo-Indians with the aboriginal tribes? When he was thinking about these problems, and I'm not going to give you in detail what the answers to these questions were or the kind of answers that they came up with, the important part is that we talk about these words, affinity, distance, similarity, and so on and so forth. At that time, there were no measures there were no statistics of measuring distance between members of two populations. In other words, you could not, based on observations of members of population groups, you couldn't actually quantify the distance between these two populations. You needed a measure of distance. And this measure of distance, because they were talking about anthropometric measurements, this measure of distance had to be based on anthropometric measurements. At that time, Mahalanobis was aware that Carl Pearson was thinking along the same lines, and he was studying various kinds of races in Europe. And he had constructed a coefficient of racial likeness and had applied that to skull dimensions of various fossils found in Burma and elsewhere. This paper was also published in Biometrica, but Mahalanobis came to a realization when he looked at the structure, the mathematical structure of the coefficient of racial likeness. At the same time, he was doing these studies on Anglo-Indians of Calcutta, and he says, I soon realized that Carl Pearson's C-squared, which is the coefficient of racial likeness, was properly a test of divergence between two samples rather than a measure of actual magnitude of divergence. This really means that, let me explain this in 30 seconds. This really means that if you have two samples of individuals from two population groups, what the coefficient of racial likeness did was to look at the divergence between these two sets of samples. It was not an estimator of the true unknown distance between the two population groups. For a true unknown, for a proper estimate of the true unknown distance, it needed to be independent of the sample size. The measure could not depend on the sample size, and that was the major problem with the coefficient of racial likeness. So this is exactly what I said. The magnitude of C-squared depends on the size of the samples, and a proper estimate should be independent of these sample size. Mahalanobis wanted to study this better, also wanted to discuss with Carl Pearson before he started thinking afresh about measurement of distance between population groups. So he went back to England in 1926-27 and spent about 6 months in Carl Pearson's laboratory at the University College in London. And he soon realized that it was really a test of divergence and was not an actual measure of the magnitude of divergence, and because it was dependent on the sample size. So he thought of his own measure, and I'm not going to give you the equations to these measures that he or the CRL, the actual mathematical formula, but Mahalanobis thought of, and he called that as the first provisional measure of caste distance, and he called that as D, and he proposed that in Analysis of Race Mixture in Bengal, and this was the title of his talk in the Indian Science Congress Anthropology section in 1925. He realized that a large body of experimental data, anthropometric data, were available on various European populations, And one of the things that he realized was that, on each individual, you're taking a multidimensional vector of observations on various anthropometric measurements, body dimensions of each individual. And what CRL, the coefficient of racial likeness, was doing is that it was not taking correlations of body measurements into account. In other words, your length of the total length of the leg is correlated with the length of the femur. And therefore, these correlations needed to be taken into account, and you couldn't treat those observations as independent observations on the same individual. And the coefficient of racial likeness was not taking that into account. So Mahalanobis thought about that and devised a measure, and that measure was called, he initially called it D, later it was refined and was published in the Asiatic Society of Bengal, called Tests and Measures of Group Divergence. And this particular formulation of the D-squared statistic, and he derived many properties of the D-squared statistic and applied them in various situations. And those are undoubtedly the most profound contributions of Prasanta Chandra Mahalanobis. He, like I said, was the father of statistical science in India. He made many seminal contributions to statistical science, but this was undoubtedly his most profound contribution, and this is globally acclaimed. You don't really have to have, you're measuring in this context, you're measuring distance between two human population groups, but you could also measure the distance between, let's say, two sets of screws produced by two separate machines. And if you wanted to measure the distance on each screw, then you would measure the diameter, maybe the length of the spirals, and so on and so forth. And based on a series of measurements, you could actually measure the distance between two inanimate objects. So this is not the D-squared measure can be applied on any multidimensional vector of observations on any unit that you want to think about, whether it's human individual, kangaroos, screws, whatever, what have you. So it's very, very widely used, and this has been a very profound contribution of Prasanta Chandra Mahalanobis. While he was doing it, I said Mahalanobis met with Nelson and Nandel, and later he also met D.N. Majumder, who was an anthropologist, and they undertook an anthropological survey of the United Provinces. The then United Provinces in late 1930s was like Uttar Pradesh, Uttarakhand, and that particular area was called United Provinces, and they undertook a statistical study of the provinces, and they came up with various kinds of statistical methods in order to depict multidimensional observations on a two-dimensional plane. Many of you are absolutely familiar with principal components analysis. So this is a forerunner of the principal components analysis that Mahalanobis had thought about, perpendicular ordinates and of correlated measurements, and therefore you needed some mathematical transformation, and those were thought through, and they were later defined as principal components. So in the UP anthropometric survey, they chose a series of populations: 2 Brahmin groups, 4 artisan groups, 8 tribal groups, and some groups that were historically or anthropologically known to be due to hybridization of Brahmin groups with artisan groups, and or between artisan groups and tribal groups, such as the Chamar, the lower caste Chamar, and a whole set of populations whose anthropological history had not been inferred. So the question that they ask is, in these cases, in some cases, we know the anthropological history. Are they borne out by physical dimensions, by a study of the physical dimensions of samples of individuals from these population groups, etc.? So what they found, or is there a correspondence between body dimensions and the social hierarchy that these population groups enjoy? They did find that among some of the groups that form the main sequence of populations, there is close correspondence between social status and resemblance to the Brahmins occupying the highest tier in the caste hierarchy. But also they found that in terms of the origins, such as the origins of the Chhatri, the Muslim, the Garyas, the Muslims of United Provinces were primarily converted Muslims, but they were as a result of hybridization between groups, and they found that there are a number of cases in which physical evidence definitely indicates possible origins contrary to accepted opinion. So based on physical dimensions, they were looking at these human, diversity, and they were trying to answer certain questions that relates social structure with body dimensions, and origins with body, inferring origins using body dimensions. And they found that in some cases there was correlation or correspondence, but in some cases there was no relationship between inferred origins and the origins that you inferred anthropological origins and the body dimensions. They were also acutely aware of the way that you could use these population nomenclature as upper caste, lower castes, etc. So they avoided using those, and they used a neutral word group, and also defined what they meant by a group. These are ethically important, that you need to use neutral words, and today it's become extremely important because of equity, diversity, inclusion, these kinds of considerations. And they defined a group as consisting of individuals who belong to the same caste, religion, or tribe, and who live in the same district. After the UP anthropometric survey, Mahalanobis, D.N. Majumder also had lots of questions about populations of Bengal, and this was undivided Bengal. This was East Bengal and West Bengal combined. They undertook an anthropometric survey of populations in Bengal, and as you can see, Mahalanobis is not an author of this particular paper. It is Mahalanobis' disciple, C.R. Rao, the oldest living statistician in the world. Today, he is over 101 years old. C.R. Rao. D.N. Majumder is the same D.N. Majumder, the anthropologist. Mahalanobis relegated to the background and let the baton be carried forward by C.R. Rao. He wrote a foreword to this particular report of the Bengal Anthropometric Survey. And in the foreword, or in the conclusions, he says that one of the important contributions is the demonstration of regional differences within a social group. That is, between individuals adopting the same caste, tribal, or religious name, but living in different geographical locations. So essentially what this means is that, if you had the same population, the same social group, let's say calling themselves as Brahmins, but they lived in two different geographical locations, their physical dimensions became so dissimilar that it would be quite difficult to actually think of them as being one social group. So it shows that, and then they concluded that it shows that a term like Brahmins of Bengal has to be used with some caution. Essentially, geographical distance actually creates a lot of difference, or over a period of generations creates a lot of difference in physical dimensions as well. This could be intrinsic. This could also be because of environmental factors, and we'll come to that in a minute. Another interesting feature is that sometimes there is closer resemblance between caste groups within a district than between individuals of the same caste group belonging to different districts. In other words, again, geographical distance plays a major role in determining how the similarities of individuals who live in close, even though they belong to the same group, but live in geographical proximity versus living in geographical isolation. Well, Mahalanobis came to the conclusion that anthropometry was really not a good set of characteristics to study population divergence and answer questions such as origins of populations. It has to be, he said, it has to be supplemented by other physical, genetic, and serological data. And he says that our experience with anthropometry has not been very convincing. It was providing equivocal answers about population groups and their relationships. And he said that blood group variations need to be looked into. So in the Bengal Anthropometric Survey, on a subset of individuals, they looked at blood groups and actually devised certain statistical methods to depict ABO blood groups. But again, soon they came to the realization that ABO blood group was just one system, one genetic system, which did not have too much of information in order to draw inferences about these population relationships or about population ancestries. Majumder, D.N. Majumder actually emphasized that they cannot give any clue to the ethnic origin or for that matter any part of the country unless these findings are reinforced by multiple genetic systems. Just to mention that, just to recall that this is what Mahalanobis said. Having said this, having realized this, Mahalanobis also had the foresight that he should set up a human genetics unit at the Indian Statistical Institute, and he did indeed set up a human genetics unit at the Indian Statistical Institute, and continued to do various kinds of population work, looking at genetic diversities of populations. And when I joined the Indian Statistical Institute, already the human genetics unit, well, it was called the Anthropometry and Human Genetics Unit when I joined the Indian Statistical Institute, and later that bifurcated into human genetics unit, which I headed, and biological anthropology unit, which was headed by an anthropologist. All right, I'll come to the last few minutes of my talk, essentially talking about human evolution using genomic data. To cut a long story short, and I'm essentially going to focus on India and the work that's been done in India, but to put this in perspective, humankind, modern humans like you and I, we all evolved in Africa. So we are talking about genomic diversity. It is really a heritage. It's a heritage that comes out of Africa, and we were born in Africa, so to say, about 200,000 years ago, and we came out of Africa about 100,000 years ago. We can only speculate why we have come out of Africa. Perhaps we over-reproduced, and there was enormous pressure on natural resources. We were hunter-gatherers at that time, and therefore, because of lack of natural resources, we probably started to explore other geographical areas in search of food, etc. When I talk about these, dates, these dates also have large standard errors. So 100,000 years could be anywhere between 80,000 years and 120,000 years. Please bear that in mind, because these dates are not absolutely sacrosanct. When we came out of Africa, after that, we started to explore other land masses, and we moved around. One of the first places that we came to after we moved out of Africa was India, and one of the last places that we went to was by crossing the Bering Strait and going and populating the New World, the Americas. So this is how we moved around, and this much of this is through reconstruction of fossil remains, as also from various kinds of genetic studies. When we came out of Africa and we went to Europe, we found that we were not alone. There were at least two other hominid cousins that were roaming around on Europe, on the plains of Europe. One was called the Neanderthals. There were plenty of them, plenty of Neanderthals who were roaming around. And we also got to know much later that there was another species called Denisovans. They were called Denisovans. We have named them. They are called Denisovans because they were found in the Denisova cave in Siberia, and there was a finger bone that was found. DNA was isolated from the finger bones. DNA was also isolated from various skulls and other fossil remains of the Neanderthals, and when we compared the DNA sequences, you find that there were such great differences that they could not be Denisovans could not be Neanderthals, and Neanderthals could not be Denisovans, so they were two separate species. What happened to them when we came to Europe? Very shortly thereafter, both Neanderthals and Denisovans vanished from the face of this planet. Did we over-reproduce ourselves, and because we were better users of tools and weapons, we actually killed them? The answer is no. Again, the answer comes from genomic studies, where genome sequences of modern humans, Denisovans and Neanderthals, have been compared, and we have statistical methods of estimating whether there has been any admixture, introgression of the genomes. And indeed, what we have found is that Neanderthals contributed to both modern human genomes and Denisovan genomes, and Denisovan genomes also contributed to modern human genomes. And essentially, this means that we all mated with each other. We were not completely isolated populations. The product of these matings, when there was a mating between, let's say, a Neanderthal and a modern human, the child of that mating, the offspring of that mating, went with the modern human groups, depleting the size of the Neanderthal group. And over a period of time, the Neanderthal group, size of the Neanderthal group depleted to such extent that they became extinct. As a result of these studies, we also know that there was a potential, so essentially they made love, not war. As a result of these genomic studies, we also know that our genome contains DNA, pieces of DNA that do not belong to us, do not belong to the Denisovans, do not belong to the Neanderthals, do not belong to us, meaning that when we compare, for example, with our ancestral populations, there are such portions of our genome that cannot be traced back to another modern human, not to a Denisovan, not even to a Neanderthal. So there are these potential unknown hominins. This was interesting, and when we started our work in India, and we did these studies on the Andaman and Nicobar Islands, the Jarwas, Ongis, the tribal populations of Andaman and Nicobar Islands, and we were looking at their origins and their population structure. Again, I'm not going to describe in details. All that I will say at this point, the Andamanese population structure is such that they have a separate ancestry compared to populations of mainland India. I will primarily focus on mainland India. These Andamanese people, we found in their genomes an unknown, bits and pieces, fragments of DNA that could not be traced back to any of the known ancestors, not even to the Neanderthals or the Denisovans. So here was among the Andamanese people pieces of DNA that needed to be attributed to a potentially unknown hominin. While we were doing this study, there was another group of people who were studying tribal populations of Melanesia, and they found that the ancient Melanesians interbred with a mysterious hominid. Suffice it to say that there was a lot of coastal migration from out of Africa into India, through Australia to Papua New Guinea, and so on and so forth, through these various island populations. We still don't know whether the mysterious population that the geneticists who were studying Melanesians, that mysterious hominid was the same as the mysterious hominid that we found in Andaman and Nicobar Island, because they have not released their DNA sequence data yet. So if they release their data, then we can easily answer this question, whether there was an ancestry or mating with a mysterious hominid or an unknown hominin who was roaming around in these through these island populations at that time, in ancient times. While we were looking at these Indian populations, etc., several of us, including Professor Brahmachari, Mithali, and groups in IGIB and elsewhere, we came together with a whole set of geneticists from various parts of Asia, and we wanted to anchor Indian genomes in the context of Asia. And let me just quickly explain. All of these are names of population groups of Asia, Southeast Asia, Japan, etc., many countries in Asia. And also, if I can explain this, graph to the left of your screen, left of my screen, essentially, this graph is that this graph is ordered in such a way that the oldest population, the most ancient populations, are to the bottom of the graph, and the most recent populations are to the top of the graph. So if we look at Indian populations, where do they belong? Are they in the top of the graph, or are they at the bottom of the graph? I've already told you that after humankind evolved in Africa, they came out of Africa and got into India, and therefore the expectation, even before we did this work, and this work is based on hundreds of thousands of DNA markers, and looking at, I will explain this kind of checkered colored picture later in my talk. Right now, I won't. I'm only focusing on this particular tree, which is called a phylogenetic tree, based on depicting relationships among population groups of Asia based on on assessment of their genomes. Essentially, even before we drew this tree, we knew that Indian population should belong to the bottom of this tree, meaning should be ancient, because human populations came out of Africa and populated India to begin with. So indeed, what we found is that there is an early divergence, and these are populations of India. We looked at a small number of populations. At that time, money was scarce, and so we did not This is 2009, over 10 years ago, 12 years ago. And there were some populations that we had sampled from North and East India. There were some populations that we had sampled from South India. And essentially, what was shown is that the ancestries of populations in North and East India was different from the ancestries from of populations from South India. Now, this was based on a small number of populations, and therefore we wanted to study this in greater detail, especially in the context of India. So again, Professor Brahmachari, who was by then the Director General, was he? Can't remember. But essentially, we wanted to look at the entire ethnic composition of India and draw inferences about the ethnic composition of India. And if you look at the anthropological classification, there are 450 tribal populations, including subcastes. There are about 4,000 subcastes and about 150 religious and migrant groups. And they have different kinds of social organizations and do various kinds of occupations. They also speak different kinds of languages, Austroasiatic, Dravidian, Tibeto-Burman, Indo-European, and so on. And I would like to point out that the Austroasiatic language of speech is essentially spoken by some tribal populations of India, such as the Santals, the Mundas, and so on. So that's exclusively spoken by the tribal populations of India. So like I said, that Professor Brahmachari essentially came up with this money, and I think there were a total of 6 CSIR institutions and the Indian Statistical Institute, and we all came together and we sampled a large number of populations from across India, and we actually scored each genome on the basis of 405 markers, and tried to find relationships among these genomes of groups of individuals belonging to these various populations on the basis of these of the data on 500 on the basis of these 405 genetic markers. And I will just show you what we reconstructed. Essentially, what these are. This is again another way of depicting a phylogenetic tree, providing depicting the relationships among these various populations. Suffice it to say that this population is colored by language groups, and as you will see that there is a strong clustering of these population groups by language. But I must also point out that in India, language and geography are confounded. People in northern parts of India speak Indo-European languages. People in southern parts of India speak Dravidian languages. In the northeastern part of India, Tibeto-Burman languages, and Austroasiatic language of speech is the fragmented linguistic family spoken in central and eastern regions of India inhabited by the tribals. So essentially, if you look at this particular population reconstruction, you find that it's clustered by language. Also, you can interpret this as clustered by geography. We then became a little bit more greedy. Technologies have evolved, costs had fallen, cost for sequencing entire human genomes had fallen, and we undertook a major project in Asia based on sequencing of genomes, and this was called the Genome Asia 100K project. Again, looking at Asia and looking at India in the that was our interest, looking at India in the context of all of Asia, primarily by looking at whole genomes as opposed to fixed points on the genome. We sampled a large number of populations, and as you can see in this map, each circle represents a population, and the size of the circle is proportional to the sample size. This is based on sequencing efforts, and we also estimated the amount of Neanderthal admixture in each of our Indian populations. Essentially, 2 to 3% of our genomes comprise the Neanderthal genomes. Less than 1% of our genomes comprise the Denisovan genomes. And also, but with respect to the Denisovan genomes, there's an interesting structure, which is that the tribal populations contain the highest amount of Denisovan genomes, even if it's less than 1% of our entire genome. It is a significant proportion, primarily because our entire genome, as Professor Brahmachari has said, is 3 billion nucleotides. And even 1% of 3 billion nucleotides is quite significant chunk of DNA. And if we move from the tribal populations to the upper caste populations, there is a reduction in the extent of Denisovan admixture in our genomes in the genomes of these populations, and that's interesting and significant. I don't have the time to actually explain in greater detail. One of the questions that we had asked is that India comprises a vast array of genetically heterogeneous populations, and they all came at different times from different places and may have different ancestries. And one of the questions that we had is how many footfalls of how many distinct ancestral types can we find in mainland India? And of course, this question was asked by multiple people, but we were beaten. We did the study when asked these questions when I was at the Indian Statistical Institute, and we completed the answer to these questions when I established the National Institute of Biomedical Genomics, primarily because technologies became available in this institute. So prior to that, the group from CCMB and the group from Harvard Medical School, they had collaborated to reconstruct Indian population history. They also asked the same question, how many ancestral types can we find? And they essentially found that there are two ancestral types, one comprising the North Indian populations and one comprising the Southern Indian populations. Now, the sampling, the number of populations sampled in this particular study was restricted. For example, they did not have any samples from Northeast of India. So if Northeast of India had a different ancestry, they would not be able to detect it. So we said that we should, our sampling was much better. So we said that we should look at this question afresh. And we did look at this question afresh. Actually, there was a considerable amount of overlap when they were doing their study and we were doing our study. And this is the result of our study. I'll give you the take-home message at right in the beginning. We estimated admixture in each individual that we sampled, and we found evidence of 4 ancestral populations in mainland India. And so let me explain this graph. On the x-axis, you have a very large number of dots that you can't even see, and I couldn't number those. These are individuals. These dots represent individuals. Each vertical line essentially represents the extent of genomes, how much of it is pristine and how much of it is admixed by genomes of other populations or other individuals. So each color represents an ancestral type, and each vertical line represents an individual, and the checkered colors essentially represent the extent of admixture that there is among in the genomes of that genome of that particular individual from different ancestral types. So if you look to the left of the screen, you find that these are pristine, almost pristine green genomes, and if you look to the right, they're almost pristine blue genomes. And if you look somewhat at 3/4 of the distance from the left-hand side, you find the sky blue, and there are some pristine genomes of the sky blue, and similarly in the center, there are some pristine red genomes. So these are individuals who are from different populations, but are essentially unadmixed. But if you look at the other regions of this graph, you'll find that the vertical lines are checkered by different colors. But overall, there are 4 different colors, and these 4 different colors represent the ancestries. So most of us have multiple ancestries, or at least genomes from multiple ancestors. There are only a few who have pristine genomes, meaning that they have unadmixed genomes from one particular ancestral types. Can we identify these ancestral types? Yes, we can. We know where we have sampled these populations from. We know what kind of languages they speak, the geographical region of sampling, etc. And we have been able to identify these 4 ancestral types as Indo-European, Dravidian, Austroasiatic, and Tibeto-Burman. So these are the 4 ancestral types that are present in mainland India. Again, I emphasize that there is a confounding between geographical region of habitat and language. North India, South India, Central India, fragmented Austroasiatic, and Northeast of India. We published this in PNAS. The data that we gathered in the Genomatia project not only informed about genomic diversity and the population structure and the origins of populations, etc. They also provide information on predisposition to diseases, response to drugs, interactions with environmental factors, propensity to infections, progressions of diseases, etc. So in other words, the genome comprises a huge lot of information, and studies on genome diversity can actually inform medical science as well, in addition to providing basic evidence of our ancestry, where do we come from, etc. I don't have the time to explain all of this, but in this particular study, in the Genomatia study, we did find some very interesting cancer predisposing genes as well. So I come to the last slide, which is my most important slide, and this is the slide that I want to the notion that I want to leave you with. And this slide, I'm going to read out this slide. Of course, I know that you can read English, but still, I'm going to read out this slide. It makes me feel good. This is a passage from a poet, African-American poet, Maya Angelou, and I'm going to read it out to you. It says, "It is time for the preachers, the rabbis, the priests, and the pundits, and the professors to believe in the awesome wonder of diversity so that they can teach those who follow them. It's time for parents to teach young people early on that in diversity there is beauty and there is strength. We all should know that diversity makes for a rich tapestry, and we must understand that all the threads of the tapestry are equal in value, no matter their color, equal in importance, no matter their texture." Thank you very much for your attention. I greatly appreciate the invitation accorded to me by IIT Jodhpur and to Professor Shantanu Chaudhary and Mitali Mukherjee. Thank you very much.